Let's talk about the neuroeffector junction. This is the synapse between the efferent and the effector organ. So the synapse between the postganglionic neuron and the effector tissue in question. And we want to compare this to a typical neuron to neuron synapse. The difference here is that we have varicosities. So this is not one neuron to another neuron. This is a neuron ending in these swellings that we call varicosities that actually have the synaptic vesicles with the neurotransmitter inside, and that will release these neurotransmitters to bind to the tissue and bring about the response, okay, by binding to these receptors. So it's very similar to the synapse that we looked at earlier, but there are some crucial differences. Let's look at the sequence of events here. So step one, the action potential arrives at the varicosity. Similar to what we looked at earlier, we have voltage-gated calcium channels along the membrane of the varicosity, and the action potential here opens up these voltage-gated channels that triggers exocytosis of the vesicles that contain these neurotransmitters. They will simply cross the junction and bind to their corresponding receptor. And then looking at the signal transduction details we described a minute ago, that is how they ultimately bring about their cellular response. And then the options for clearing the junction is we can degrade the neurotransmitter, we can reuptake it back into the varicosity and repackage it to a new vesicle, or it can simply diffuse or move away from the junction. Now, I do want to point out, because we have two different branches of the autonomic nervous system, the sympathetic and parasympathetic, we're going to see two different types of neuroeffector junctions. On the postganglionic parasympathetic side, we have a postganglionic neuron interacting with an effector cell. And remember, the acetylcholine released here binds to a muscarinic type of cholinergic receptor. So this is going to be a metabotropic, but it's going to be binding acetylcholine only. This is the parasympathetic side. On the sympathetic side, the neurotransmitter is different, right? It's catecholamines, namely norepinephrine. And so that will bind to adrenergic receptors. So this can be many of the variations we looked at, alpha receptors, beta receptors, depending on where this tissue is located. And then the options for clearing the junction here are going to be a little bit different, because we can repackage these into vesicles, but the mitochondria here is just indicating that because it's a different neurotransmitter, it's going to be made differently than acetylcholine. It's actually going to be made by monoamine oxidase. Monoamine oxidase in the mitochondria is responsible for making norepinephrine. And so that's another sort of fundamental difference across these two neurons. Okay. So all right, let's move to the third and last objective here. Describe how the central nervous system regulates the dual innervation of the autonomic branch of the peripheral nervous system. So we're going to kind of zoom out. We've been kind of looking at the cellular details, and we're going to zoom back out and look at this more holistically. How do we have this dual innervation, this comparative secrecy, and how does this bring about cellular responses in both the sympathetic and parasympathetic state? So let's look at how the CNS helps to regulate this. So we discussed the dual innervation of organs. This simply means that all of our effector organs, salivary glands, lacrimal glands of the eyes, heart, kidneys, muscle, all of those organs are being innervated by both divisions all the time. The balance is typically what keeps us at rest. So when we're at rest, there's somewhat of a balance, but the parasympathetic system is slightly more predominating. Okay, so as I'm sitting here in somewhat of a restful state, there's both of the systems innervating my organs, but parasympathetic is slightly more predominating in terms of its influence in the resting state. When you're excited, the sympathetic is going to predominate. So let's say you're exercising, let's say you're stressed, let's say you're walking, really briskly. The sympathetic side of things will have a more predominance versus parasympathetic. And then when we think about the regulation and the change, let's say you go from a resting state to a more excited state. There's going to be a dip in the parasympathetic activity. Likewise, that's going to be coupled with an increase in the sympathetic activity. And the opposite is true. Let's say you go from being chased by a bear or a dog, and you end up calming down, relaxing, going back to a restful state, then you're going to see a drop in the sympathetic tone and then an increase in the parasympathetic tone. So that is comparative secrecy. As one branch decreases its influence, the other branch will increase its influence, vice versa. Let's look at an example here of blood pressure regulation regarding the autonomic reflex, and just look at a practical example of how those changes come about. So let's say you go from a seated position to standing up very quickly, okay? What does the autonomic reflex in your body look like to control your blood pressure at this time? So when you stand, gravity is going to pull all the blood, not all, but most of the blood in your body down to your legs. And so that pooling of blood has a decrease in blood pressure response to the rest of your body. So your legs are going to have increased flow, but areas like your trunk, your arms, your head and neck, they're going to receive the signal as if there's a drop in blood pressure or a drop in perfusion to those other areas. The visceral receptors in those areas will detect that change in blood pressure, and they're going to trigger a response by sending signals to the central nervous system. So they're going to send signals on afferent pathways, and the way that they send those signals is a decrease in the frequency and action potentials. If you recall us describing frequency coding means that the way the nervous system grades signals is by altering the frequency of action potentials. So to send a signal that there's a drop in blood pressure is a drop in the frequency of action potentials on the afferent fibers toward the central nervous system. Now, when these signals get to the CNS, namely the medulla oblongata in the brainstem, they're going to receive those signals, process that information, and come up with the instructions for a corrective change that's going to alter the change and result in an increase in blood pressure. So the way that this happens is the medulla sends signals on efferent fibers. Remember, afferent is sensory going to the CNS, efferent is motor going towards the peripheral nervous system. So the afferent signals are going to do two things. They're going to drop the parasympathetic tone, and they're going to increase the sympathetic activity, right? Drop the parasympathetic activity, increase the sympathetic activity, and that is going to result in the changes on heart, on blood vessels. The example here would be to decrease the vagal stimulation to the heart and increase the beta-1 stimulation to the heart, okay? So that will help to result in bringing about an increase in blood pressure, an increase in heart rate, and an increase in blood pressure, okay? But notice how the effect had to result in changes to both parasympathetic and sympathetic systems. Now, this will result in obviously a negative feedback when blood pressure rises. This goes back to the initial change, and this is going to close that feedback loop as the error signal is now turned off, right? If you remember that from homeostasis. So this is an autonomic reflex, an example of a homeostasis loop, and this is how the autonomic system can bring about those changes and can help to maintain homeostasis. Now, looking at the overall parasympathetic activity, this is the quiet state, the relaxed state. This is involving resting, digesting. So you're going to see increased glandular secretion. You're going to see increased GI motility, gastrointestinal motility. This is going to help you absorb food. This is all going to be involved in the relaxation state. We're also going to see a drop in heart rate and a drop in blood pressure as well. This is why if you've ever heard of the vagal stimulation, right, the sleeper's hold, when you stimulate the vagus, the carotid bodies and the vagus receptors, you can actually make someone pass out, like put them into a sleeper's hold, or you can stimulate their vagal activity so much, their heart rate drops, their blood pressure drops, and they can actually pass out because of decreased perfusion to the central nervous system, to the brain. So that's sort of an example of how the parasympathetic activity works. On the opposite side, we have the sympathetic activity, the fight, flight, freeze is another type of sympathetic response. This is preparing you for emergency states, stressful states, exercise. And when we say stress, this can be physical stress, right? Actual stress to your body. Let's say surgery is an example of physical stress, or physiologic stress. Okay, stress, let's say you're dehydrated, that's physiologic stress. Or it can even be mental stress. Let's say you're worried about an exam coming up, or you're worried about financial, stress. Those are different sort of categories of stress, but they all can elicit a sympathetic response. Okay, this is why post-pandemic, a lot of clinicians recognize that because of the events of the pandemic, people were usually more in a stressful, sort of sympathetic, sort of, just an overall sympathetic state because of those events. And that led to a lot of other health consequences. And so I just want to point out here that stress can be multiple types and not just like actual stress. The results here are going to be increased heart rate, increased blood pressure, mobilizing energy stores so that those can be utilized to help you fight or flight or flee. And then the dilation of pupils. The function of dilation of pupils is interesting. And again, think about this evolutionarily. If you are in the presence of a threat, let's say you see a bear, you need your pupils dilated to be able to take as much light in to be able to perceive your immediate surroundings and detect any additional threats. So dilation of pupils is a sympathetic response, whereas pupil constriction is more parasympathetic. And then decreased gastrointestinal and urinary functions. This is usually why folks will sometimes lose control of their bladder, have an accident when they are in a stressful situation, when they receive stressful information, and that can be because of an automatic collapse of the parasympathetic influence and the sympathetic influence, which decreases urinary and gastrointestinal control. When you think about both branches, again, both are active at rest, but parasympathetic is just a little bit more predominating than sympathetic. And then looking at the areas in the brain where these control centers live. So we have cardiovascular control centers in the brainstem, in the pons and the medulla, and then we have respiratory control centers in the brainstem as well. So these two areas of the brainstem will receive signals and then bring about the corrective response that can change our cardiovascular function, blood pressure, heart rate, or our respiratory function, which is our breathing rate, respiratory rate as well. And then the hypothalamus is another interesting area. The hypothalamus is what brings the connection to our emotional states, right? This is how, again, stressing over a task, stressing over finances, any other type of sort of emotional stress that happens can also result in activating these cardiovascular centers. So you can be stressing over something that's not physical, but still have a very visceral and physical response in your body. All right, lastly, here are just some helpful study tables. Again, nothing to memorize, but just to show the response of each division of the nervous system here, peripheral nervous system, on different systems. So on the heart, specifically on the SA node, AV node, we'll talk more about this in our final chapter in the cardiovascular system, on the lungs, on blood vessels. What is the effect brought about on the parasympathetic and sympathetic side? And then what are the actual receptors that are involved on the sympathetic side as well? So a nice maybe study tool or a reference tool, but not necessarily anything to memorize in detail.